Swayam Prabha. Digital India. Educated India.
Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different properties of the enzyme in the course Enzyme Science and Technology. So in this context, in this particular module, we are discussing about the different approaches and the way in which you can be able to design the inhibitor for the enzyme. Now, what we have discussed about the traditional approach, where you are actually going to have the no information about the enzyme structure or the inhibitor structure. What you require is you require the enzyme assays, and you can be able to screen the different types of inhibitors from the different libraries. One of the major drawback of the traditional approach is that it will not allow you to improve the inhibitor, because you don't know the structure of the inhibitor, so you don't know which region of the inhibitor is inhibiting, or which region of the inhibitor is important for the interactions. To overcome this particular problem, people have started looking at the targeted approach. So in the targeted approach, you actually will know the structure of the enzyme, you actually will know the structure of the inhibitors, and that's how you can be able to use the different types of computational approaches to to see how the inhibitor is binding into the binding pocket and what are the different interactions are involved. Based on these approaches, you can be able to use either the ligand based approach, where you can actually know that this is the ligand what actually fits into the active site, and then you can take that ligand and search the similar ligands into the database, and that's how you can be able to identify some of the potential inhibitors. Or you can also be able to use the receptor based approach, where you can actually be able to study the receptor binding site, and you can, based on the binding site, you can be able to identify the crucial interactions which is responsible for the receptor to interact with the ligands, and considering and keeping those interaction intact, you can be able to design the new ligands. Both of these approaches we have discussed in detail in the previous lecture, and we have also taken some of the case studies how you can be able to use some of these approaches. Now in today's lecture, we are going to discuss about the computational approaches, how you can be able to use them for designing the new inhibitors. So when we talk about the computational approaches, it is well understood that you actually know the structure of the enzyme, and you also know the structure of the inhibitors. If the structure of the enzyme is not known, then you actually have the possibility of designing enzyme structure either by the homology modeling or the de novo modeling. So prerequisite of the computational based approach is that you should have the enzyme structure and as well as the inhibitor structures. So in a structure based drug design or structure based inhibitor design, what you require is you require a database of the ligands, so that you can be able to select the different types of ligands from the database and you can test them into the computational approach. You require a target protein, and you also should know the structure of the target protein. Not only that, you should also know the position of the active site and the binding pockets and all that. Okay, and then you can actually be able to put them into a process which is called as the molecular docking. So within the molecular docking, what it is going to do is it is actually going to see how the ligands are fitting into the active site, and the ligands are docked into the protein's active site, and the pharmacokinetics and the pharmacodynamics optimizations are actually going to be done so that you can be able to know how well these ligands are fitting into the active site. So the question comes, what is docking? So what is docking? So docking refers to the ability of to position a ligand into the active site or a predefined designated site of a protein and calculate the specific binding affinities. It means it is actually going to tell you about the affinity constants. Then docking algorithm can be used to find the ligand and the binding conformation at a receptor site close to the experimentally determined structures. Dogging algorithms are also used to identify the multiple proteins to which a small ligand can be used. You have some of the important docking programs such as Gold or Autodocs, Ludi, and Hex, and some of these programs are free, and whereas some of the programs are the licensed programs, so you can actually be able to use them for performing the docking. And what docking is exactly doing? Docking attempts to find the best matching between the two molecules. Remember that the docking is actually works on the lock and key model. So remember that lock and key model, right? So lock and key model says that there is a lock and there is a key, right? So if there is a lock, there is always a definite key, right? So if you can be able to make the match between the two molecules, whether it is a lock and key model or whether the induced fit model. So it includes finding the right key for a lock, right? That is the correct statement, okay? Given the two biological molecules, you can actually be able to determine whether the two molecules interact with each other. If so, what is the orientation that maximizes the interaction while the minimizing the total energy of the complex? Because these are the two important parameters. One, fitting, and the second is the free energy, right? So it should be have the total free energy of the molecule lower, and it should have the best fitting. And the goal is to able to search a database of molecular structures and retrieve all structures that can interact with the query structures. So the aim of the molecular docking is to achieve an optimized conformation for both the receptor and the ligand, and the relatively orientation between the protein and ligand, such as the free energy of the overall system is minimized. Successful docking methods search high dimensional spaces effectively and use a scoring function that correctly rank the candidate dockings. So you have a target molecules, you can have the ligand molecules, and with the help of the molecular docking, you will be able to say whether this particular ligand is fitting into this particular active site or not. So what is the importance of the molecular docking? Okay, so molecular docking is actually going to identify the ligand's correct binding geometry or the pose into the binding site or the binding mode, whereas it is actually going to predict the binding affinity or the binding scores, right? So it's first of all, it is actually going to say at what position this particular ligand is actually going to go and bind into the target, whereas it also going to tell you the scoring functions. So it's actually going to tell you the affinity parameters, and that can be used for the comparing the molecules. So that can be used for comparing the molecules, and that's why the molecular docking can be used for rationally designing the target molecules. Now, when you talk about the molecular docking can be performed in two different modes. So you can actually follow the lock and key model, or you can actually be able to follow the induced fit model, which means you can actually be able to do the rigid body docking, or you can be able to do the flexible docking. So in the rigid body docking, the internal geometry of both the receptor and ligands are treated as a rigid. So in this case, you are actually going to follow the philosophy of the lock and key model. Whereas in the flexible docking, you are actually going to have the induced fit model. So an evaluation on the rotation of the one of the molecule is performed. Every rotation, the energy is calculated. Later, the most optimal course is selected. So in this case, you are actually going to make one of the molecule as the flexible molecule. So either you can actually be able to use the ligand as a flexible molecule or the receptor as a flexible molecule, and that's how you can be able to do the flexible docking. Majority of the time, it is ligand what you are actually going to make as flexible molecule. Now, as far as the docking is concerned, you can actually be able to do the docking between the different pairs. You can actually do the protein ligand docking, you can actually do the protein protein docking, and you can actually be able to do the protein nucleotide, or I'll say DNA or RNA docking. Okay, so this is the protein DNA RNA docking. So either of these pair can be used and can be studied in the docking experiments. Now, when you want to do a docking experiments, you are actually going to follow the different types of workflow. So first is number one, you are actually going to do the target selection, which means you are actually going to select the enzyme, and not only the enzyme, you are actually also going to select the binding site. Okay, and there are multiple procedures and multiple steps how you can be able to select the target. Once you have selected the target, then you are actually going to prepare the target. Okay, this means you are actually going to have the different types of procedures. So you can actually be able to add the water molecules, because remember when we were talking about the protein structure, we said that the protein is always been present in a hydration shell. So you are actually going to add the water molecules so that you are actually going to mimic the real examples. And then you're also going to add the charge and other kinds of procedures, and that's how your target is actually going to prepare. On the other hand, you are actually going to prepare the ligand molecules. So you're going to select the ligands, right? So you can actually, when you want to select the ligands, you can actually be able to select the ligands from the different types of libraries. You can actually be able to use from the phytochemical libraries, or you can actually be able to use the chemical libraries. For example, you can actually be able to choose the molecule from the zinc database or the phytochemical database like the Duke's database. Once you selected the ligands, then ligand is also going to prepare. So exactly just like the target preparations, you are also going to prepare the target preparation, ligand preparation. So in the ligand also, you are going to add the charges, you are actually going to add, or you are also going to define the rotatable bonds and all that, and so on, because all these are you're going to do the flexible docking. And then you are actually going to put it into the molecular docking. And what the molecular docking is going to do is it is actually going to predict the different poses of the ligand within the target proteins, and that's how it is actually going to give you the information about the binding affinity of each target or each pose, right? Taking these into account, you can be able to do the analysis. You can to evaluate the docking results, and you are actually going to see the binding conformations, and you are actually be considering these binding conformations. You can be able to select the right pose and right conformations. So to explain all these procedures, we have prepared a small demo where we are actually going to use a software which is called as Autodoc, and where the students are explaining how you can be able to do the target selections, how you can be able to prepare the target, how you're going to prepare the ligand and all that, how you're going to prepare the grid parameter files, how you're going to prepare the docking parameter files, how you're going to analyze the results, and so on. So in this demo, the students are explaining you different steps of how you can be able to perform the docking with the Autodoc.
Welcome to the demo on protein ligand docking using Autodoc tools. So in this demo, I will be explaining the steps involved in docking of a ligand to a receptor using Autodoc tools. So I will start with a flowchart of the steps involved. These are the 10 steps involved in docking. So I will be explaining each step in detail, and parallelly I will be performing each step on screen and explaining them. So we'll start with the first step, that is procuring the receptor and the ligand. So the receptor can be, its PDB structure, its 3D structure can be downloaded from the protein data bank site given here, and it should be downloaded in PDB format. And the second thing, if the structure of the receptor is not known, then we can predict it by modeling the structure using various softwares like Modeler, Raptor X, or Fire2, etc. Now, for the ligand, we can download the ligand from PDB as well as from various other public databases like Zinc database, SpiderCam, PubChem, NCI, and various others are there. And we need to save the ligands in SDF or Mol2 format. So after procuring the receptor and ligand, we'll move to the next step, where we need to identify the binding site where we want our ligand to dock into the receptor. So for that, if the binding site is not known, then again we can use various softwares like iTaser, etc., for identification of the binding site in a protein structure. And if there is a complex of the ligand with the receptor already available, then we can extract the residues which are involved in the interaction. So I will be showing how to extract those residues using PyMol. So I will open PyMol here. So in PyMol, we'll go to File, and here we will open the ligand bound structure which I have already downloaded from PDB. So here I will be, this is the structure which I have downloaded. So here we can see that the ligand is bound to this receptor here, and now we need to know the residues which are involved in the interaction. So here in the right panel, we will go on, in front of all it is written as A, where we will click A means action, and here we will go to preset and then click on ligands. So it will zoom up the ligand on screen along with the along with the bonds and interactions. So now we need to label the residues so that we can know what are the residues involved. So here in front of this receptor name, we will click on L, that means label, and we will click on residues. So it will label all the residues. Now again we will go to all, and here we will click on H, that means hide, and then click on ribbon. So it will hide the residues which are not involved in the interaction. Now we can just rotate this structure and note down all the residues which are showing interactions. So I have already noted down the residues which are showing interactions. So these are the residues involved in the interaction of this ligand to this receptor. So now we will move to the next step, which is um, the preparation of the receptor for docking. So before going into autodock, we need to minimize the energy of the receptor so that it shows better binding. It is in its most stable state. So now we can do the energy minimization of the receptor using Swiss PDB viewer. So I will open Swiss PDB viewer here. So this is the Swiss PDB viewer window. Here we will click on File and then open PDB file. So here this is our receptor. I will open this structure here in PDB. Uh, this it will show a pop-up. We can close this, and then we can go on select and then click on all. So it will select the complete molecule, and then we can just press Control N, which will perform the energy minimization of this structure. So after pressing Control N, here it will show the progress of the energy minimization process, and then after the process is complete, a pop-up will come up showing the energy of this the global energy of this receptor molecule. So we will see the global energy of the receptor molecules here. So here in this new window which opened up, at the right bottom it will show the energy of this receptor, which is -32054. Now we have to repeat this step. We have to press Ctrl N again so that it is further minimizes structure. So I press Ctrl N, again it is showing the progress, and then we have to compare the energy of this new layer with the previous layer. Oh, and we have to keep doing this control repetitively. We need to press Ctrl N until the energy difference between the two consequent layers is less than 100. So here the next. So after performing several energy minimization steps, now we can see the energy difference between this layer and the previous layer is less than 100. So now we can just click on this layer, and now we will click on File and then Save and then Current layer, so that it will save the most minimum energy layer of this receptor. Now we can just give its name receptor EM, that means energy minimized dot PDB. I will save this. Now we can close this Swiss PDB viewer, and after the energy minimization of the receptor, we need to prepare this in Autodoc. So we will open Autodoc now. Now we will open our energy minimized receptor in Autodoc. We will go to File, then Read Molecule, and then we will select the receptor, energy minimized receptor from the same folder. So I will select this receptor em.pdb and open it. So the receptor will be displayed on the Autodoc window, and then after that we will go to edit, and then we will delete water, so that all the water molecules are deleted, and it will not interfere in the interactions. And then again we will go to edit, and then go to charges, and we'll click on add Coleman charges. So it will show how many Coleman charges have been added. Just we'll click OK, and after that we have to go on edit, and again we have to go on charges, and then check totals on residues. So it is showing there is two residues with non-integral charges. We just have to click on the spread charge deficit over all atoms in residue. So it will spread all the charges and neutralize it, and then we will dismiss it, and we'll again check the total charges on the residue, check totals on residue. So it should show no residues with non-integral charges found. So we will click OK, and after that we will again go to edit, and then MISC miscellaneous, and here we'll click on check for missing atoms. So it should show zero residues with missing atoms. So now we'll click on dismiss, and after that we'll again go to hydrogens, and then we'll click on add, and here we have to select polar only, and in the method it will be no bond order, and in the renumber atoms to include new hydrogens, we have to check yes, and then we'll click OK. So after that we will save our receptor in PDB-QT format. For that we need to go on grid, and then macromolecules, and then choose, and then we'll choose this receptor. So it will show the some pop-up. We have to just click OK, and then a save dialog box will open. Then we have to go to the same folder and save this in save the receptor, prepared receptor in PDB-QT format. So it's already written here, receptor em.pdbqt. We just have to click save. So now our receptor is prepared. We'll go to edit, and then we'll go to delete, and delete all molecules. Now we can minimize the Autodoc window. Now the next step is preparation of the ligand. So this is the next step. Again here we have to first minimize the energy of the ligand, and then prepare it in Autodoc and convert it in PDB-QT format. So for energy minimization of the ligand we will be using Chem 3D Pro. So here I have opened Chem 3D Pro. This is Chem 3D Pro window. Here we'll go to File, and then Open, and in the same folder is open here. So we'll select the ligand.mol2 file here. So this will open up the ligand here. You can see the ligand is displayed on the screen. After that we have to go to calculations, then mm2, and then we have to click minimize energy. So a pop-up will open up. We have to just click run with these default parameters. So the energy minimization process will start. We can see the atoms and bonds moving in space, which shows that the energy minimization step is in progress. So after the energy minimization is done, here in the lower panel it will show calculation completed. Alongside it, it will show the energy of the ligand, the minimized energy of the ligand. So it will take few seconds, 10 to 12 seconds to energy minimize. That is depends on the size of the molecule as well. So we have to wait for a few seconds, and then we can save the energy minimized structure of the ligand as well. So still the energy minimization step is running. So for energy minimization of small molecules, Chem3D Pro is better, and for bigger molecules like a protein molecule, that Swiss PDB, which I showed earlier, is considered better. Now you can see that still the energy minimization is running, and the atoms and bonds are getting arranged in their minimum energy conformation. Okay, now we can see here in the lower panel, it is showing calculation completed along with the energy of the ligand. So now we'll save it. We'll go to file. We'll go to save as, and here we will just change the name of the ligand so that you can have a different file. I will write ligand em.mol2 and then save it. So the energy minimization of the ligand is done. After that, we have to prepare the ligand in the we have to prepare ligand in Autodoc tools. So for this, again we will open Autodoc, and here and in Autodoc we will go on ligand and then input and then click on open. Then we will navigate to the folder where we saved our energy minimized ligand and open it in Autodoc. So this is our energy minimized ligand in Mol2 format. I will open it in Autodoc. It will show the ligand summary. We will click OK. And after that, we'll go to ligand again and then go to output. In output, we will click on save as PDBQT. And now we will save this ligand in the same folder in PDBQT format. So it's already written as ligand em.pdbqt. We'll just save it. Now again we will go to edit, delete, and then delete all molecules. Now the ligand preparation is also done. Now we will move to our next step, that is the preparing the grid parameter files. It contains the information about the coordinates of the grid, and that grid will define where we need to dock our ligand. So we will put our grid at the site which we have extracted from the PyNode. So again, for this we'll go to the Autodoc. We'll open Autodoc here, and now we will go to grid, and in grid we'll go to macromolecule and then open. So we'll open our macromolecule saved in PDBQT format, that is receptor em.pdbqt. We'll select this and open it here. So we'll click no in the pop-up, and then it will show, it is showing like we need to correct some charges on the residues. That will be done later. We'll just press OK, and after that we'll again go to grid, and then we'll go to set map types, and then we'll click on open ligand, and here we'll open our ligand in .pdbqt format from the same folder. So this is ligand em.pdbqt format. We'll open it. So the ligand has opened up here, and now we'll move to the, again go to the grid and click on grid box. So here this window here we can define the coordinates of our grid box. So for this, first what we will do, we will select the residues which were involved in the interaction. So we will just open this window, and then we will select each residues one by one. So here you can see we will start with glycine 12, and then threonine 13, threonine 14, tyrosine 15, then it is glycine 201. So this is glycine 201, glycine 202, and similarly we will select all the residues here. So, so when we select these residues will be selected, will be displayed in yellow, so that we can easily adjust our grid box to contain all the residues in the grid box. So now glycine, glutamine 268, lysine 271, arginine 272, serine 275. Then we need to go to 338 now. So this is glycine 338, glycine 339, arginine 342, and then isoleucine 343. And the last residue is ASP 366. So this is ASP 366. Now we will maximize this. Now we can see the residues which we have selected is displayed in uh, yellow. Now we can just with the upper sliders we can move the size, we can adjust the size of the grid box, and with the lower sliders we can just move the grid box, we can change its coordinate. So first we will move the grid box towards our binding site, so that all the residues are contained in this. Now we will just increase the size all three dimensions. So this is now I will use the green slider, so this will increase it vertically, and then we can just rotate this molecule like this, and we can increase this size. So we can see we can rotate this in all direction and just see all the residues are contained in the grid box or not. So here we can see it is there. Now we can just move our grid box little this side, because it is containing some unwanted residues as well. So I will move them to this side. Now we will again rotate the molecule and see the grid box has moved too far. So we'll just bring it back. Using the red slider. We'll try to keep the residues in the middle, so that we can adjust it properly. So now we'll just reduce the size of the grid box, so that it doesn't contain the unwanted residues, and again rotate. You can see this side some residues are going out, so again we will increase the size, and we can move it little this side. So now we can see the residues are almost inside. We can increase this little bit more, and then you can just rotate it in all directions. You can see all the residues are inside only. You can reduce the size this way a little bit and then move it. Now you can see all the residues are inside the grid box. So we will just go to file and close saving current. So the uh, grid grid parameters we have set, and now we need to save this grid parameter. So we will go to open grid, and then we'll go to output and then save GPF. GPF means grid parameter file. So we need to save this in the same folder where we have saved all other files. We can write it like grid dot gpf. It is important to write dot gpf in this file name. Now I will save this, and after that again we'll go to edit, then delete, and then delete all molecules. So it will delete everything. Now we will go to the next step, which is the preparing the docking parameter file. So now we'll prepare the docking parameter file, that is .dpf file. So for that again we will go to autodock, and then we will go to docking here, and then in docking we'll go to macromolecule and that set rigid file name. So again we will go to the same folder, and here we will save in for rigid file name. The receptor in this docking is rigid, that means it will not change its conformation. So we'll select the energy minimized receptor and then open it, and nothing will be there on the screen, but it's fine. Then we will go to docking and then ligand and open. Then we will open the ligand from the same folder in .pdbqt format. So it will open up a pop-up. Here we need to, and it will also show the ligand on screen. So here we will press on accept. After that we will again go to docking, and in docking we will click on search parameters. In search parameters we will click on genetic algorithm. Here it is. Here we can set the GA runs, like how many conformations of the ligand we need the autodock to create and then dock. So it is set to 10, it can be set to 50, 100, depending upon the requirement. Then we will press accept, and then after that we'll again click on docking, and then we'll click on docking parameters. It will open a pop-up. We have to just click on accept, and after that again we will go to docking, and then we will save the files. We will click on output and then click on Lamarckian GA, and we will navigate to the same folder, and here we will save the file in .dpf format. So I will write docking.dpf. It is again important to write the .dpf so that it will be easily read by the autodock. I'll save it, and after that again we'll go to edit, and then delete and delete all molecules. And then now our preparation part is done. So we'll go to the next step, that is generating the docking job file through Raccoon. So we need to run Raccoon here. Raccoon is another software which comes embedded with the autodock. So to open this Raccoon we need to go to command prompt. I will open command prompt here. I will type cmd, and so this is the command prompt window. So in command prompt we have to go to the folder where we have saved the our where the autodock is installed. So here I will go to that folder, I press cd dot dot, and then I will write cd space in brackets. I have to write the path of. So this is the command prompt window. Now first we need to, the active folder is different, so first we need to go to the C drive and then open the folder where our Autodock is installed. So I will type cd dot dot and then again cd dot dot. So now C drive is active. Now I will go to the that folder. So you have to type the exact pathway here, cd space braces on program files x86. I will close the braces and press enter. So now the program files folder is active. We'll move to the MGL tools. We need to type the correct spelling and correct pathway, otherwise it will show some error. And then after this, we'll the raccoon is embedded in this folder only. So just we will type Python space raccoon. You need to type the correct spelling of raccoon.py, then and then press enter. After pressing enter, a raccoon window will open up. This is the raccoon window. Now here we will perform our docking. So first we will select the ligand, we'll go to add ligand and we'll select the ligand here from the same folder. So this is our energy minimized ligand in PDBQT format. So I have added that, and then we'll click on receptors, and in receptor we will add receptor file, and then we will go to the same folder and then click on our receptor file and open it in here. So it will add the receptor file in raccoon. Then we will go to maps. In maps we need to load the our GPF grid parameter file. So I will click on load GPF template, and again in the same folder where we have saved, here it is grid.gpf is there. We'll just add it here. And after that we'll go to docking, and docking will click on select docking setup from template, and here load dpf template. Now here we will select the dpf file, the docking parameter file which we created, which is in the same folder. So now the dpf file is also open. Now we will click on vs generation. So it will open up another window. Now here we need to set the directory where the output will be saved. So we will set directory, we will choose the same folder here also. So we will go on desktop, and desktop will go to the folder which we created. So this is the docking demo folder. I'll select that folder. It will say that folder is not empty. That is not to worry. We'll just click on yes. Now we will click here on generate. So it will generate a job for docking. It will show a pop-up. One docking job has been successfully generated in this folder. We click on OK, and then now we can close the raccoon here. And after that, we'll go to that folder, and then here we can see a new folder has been created as receptor em. Again here we can see one folder receptor em underscore receptor em, that means ligand em underscore receptor em, that means this ligand is being docked to this and the output will be saved in this folder. And one more file is there, run vs.bat. We have to click on this file, and then a command prompt will open up, and then it is saying press enter to start. Just we need to press enter and the docking job will start here. And now we have to wait for some time so that the docking is completed. See here our docking is completed. So now we will go to the analysis of the dock complexes. For that we will open Autodock, and here we will go to analyze and then click on dockings and open, and then we will move to the same folder. Here you can see .dlg file has been created. We will open this file in Autodoc. So it is showing read 10 docked conformations from the ligand receptor .dlg. We'll click OK, and after that again we'll go to analyze, and on macromolecule we'll click on open. So it will automatically open the macromolecule we have selected, and after that again we will go to analyze conformations and click on play. So it will open another pop-up which will be used later for saving the complex. After that again we will go to conformations and click on load. So here it will show the binding energy of the conformations. So this is the conformation with the lowest binding energy, that is minus 2.2. So now we will again go to analyze and click on clusterings and click on show. So it will open up a graph that will show the graph between number of conformations and the binding energy. So number of conformation in each clusters and binding energy. So here we can see the cluster with the lowest binding energy is having only one conformation, whereas this cluster, this is having three conformations. So now we will save the conformation with the lowest binding energy. So for that we need to select this conformation with lowest binding energy first and then click on this icon. So it will open up another window. Here we will click on write complex, and then we will save this in the same folder. So here I will write C1, that is complex 1, and then click on save. It will be saved in .pdbqt format. So now what we can do, we can open the folder which we created, and then here uh, we can open the new folder created by Autodoc, and here we can see the .dlg file. We can open it in Notepad and see the binding energy of the list of binding energy of all the conformations. It will be at the last. So this is run 5, then run 6. After run 10 it will show the table of binding energies. So here it is. This is the table showing the binding energy, the run number, the mean binding energy of the cluster, and the cluster, how many conformations are there in each cluster. So this we can just copy and paste in Excel or in Notepad for our reference. I will close this .dlg file. Now we have analyzed the binding energy of the receptor of the complex. Then the next step is the visualization of the receptor ligand complex. So this can be done by various softwares like Pymol, Ligplot, or Discovery Studio. So here I will be showing it in Discovery Studio. So I will open Discovery Studio here, and then I will go to File, then Open. Then I will open the C1 which we saved. So it will open in Discovery Studio. So here we can see that the ligand is bound here. This is the ligand. So we can click on this ligand to select it. It will be displayed in yellow, and then we will click on define ligand. So it will zoom the ligand here, and then we can click on ligand interactions. So it will show the ligand, the amino acids which are interacting with the ligand. Then we can just double click on residue and again double click so that all the residues will be selected. Then we will right click, go to labels, and then click add, and here in place of atom we can select amino acid, I will click OK. So it will label the amino acids which are involved in the interactions. We can go to interaction options here, and then it is showing what are the interactions present, like hydrogen bonds, electrostatic and unfavorable bonds. Here we will click on show distance, so it will show the distances of the bonds. Now we can analyze this, like which type of the green bonds are hydrogen bonds, and the orange ones are electrostatic bonds, and the red ones are few unfavorable bonds also. Because of these unfavorable bonds, we find the binding energy is higher, that is only minus 2 point something. So now we can save this structure as well. We can take the image from here, and then we will go to file, save as. We'll select here as image file, and we can give a file name to it, and then save it in the same folder. So this is all with docking of a ligand with receptor. So thank you.
So I hope that you have understood the different steps, and students have very in a detail they have explained how you can be able to perform the different tasks to perform the docking. Now, what are the usage of the docking, right? You can actually be able to use to develop the target, right? So you can be able to define the drug targets. You can be able to study the protein ligand interaction, which is very difficult to perform under the non-silico conditions. You can actually better understand the machinery of the life. So you can actually be able to understand the enzyme inhibitor class. You can actually be able to identify the antigen complex and other type of complexes. You can actually be able to use the protein therapies. So you can actually be able to define the different types of targets. You can actually be able to use the molecular docking to even engineer the proteins, and that's how you can be able to use them for a more efficient working. Although the reliability of the docking method is not so high, they can be provide new suggestions. So they can actually, one of the major idea is that actually going to reduce the time, and the false positive rate can be reduced using the several scoring functions in a consequence scoring system. So what are the applications of the docking? So applications are, first application is that they are going to be used in the virtual screening. So docking with a scoring function can be used to quickly screen the large database of protein drugs in silico to identify the molecules that are likely to bind the protein target of the interest. And this is actually going to reduce the time. It is actually going to reduce the, it is actually going to make the cost effective, and it's going to answer a lot of questions, because in a single day you can be able to screen 10 different types of targets. You can actually be able to screen the thousands of molecules to know whether these molecules are, what is the probability of these molecules, and that you can do in a virtual screening. Number two, you can actually be able to use the drug discovery. So you can, if you suppose you have a lead, then you can actually be able to do the lead optimizations. So docking can be used to predict in where and which the relative orientation of a ligand binding into the proteins. This information may be in turn be used to design the more potent and the selective analogs. So drug discovery is also a very important aspect of the docking. And then the third is bioremediations. So protein ligand docking can also be used to predict the pollutant that can be degraded by the enzyme. So by the bioremediations, you can actually be able to select the pollutants which you are actually going to be targeted by the enzyme, and that's how you can be able to use the specific enzyme to reduce the level of that particular pollutant. And this is very relevant when we talk about the applications of the enzyme. So when we talk about the application of enzyme, you will understand that there are so many enzymes which are available to reduce the level of pollutants. So what we have discussed in this particular module, we have discussed about how you can be able to design the inhibitors. What we have discussed that enzyme has definite areas to bind the substrate or the inhibitor, and that area is called as the active site. In some cases, you can also have the allosteric site where the enzyme is also going to interact and inhibit the enzyme. And, and as far as the inhibitor designing is concerned, we can have the multiple approaches. You can have the traditional approach where you are actually going to use the enzyme assay as a screening criteria, and you can be able to test the different types of inhibitors, and that's how you can be able to select the inhibitors which is inhibiting the enzyme. Apart from that, you can also have the targeted approach where you can actually have the ligand-based approach, receptor-based approach, or the computational approaches to design the different types of inhibitors. So with this, I would like to conclude my lecture. In our subsequent lecture, we are going to discuss some more aspects of the enzymes. Thank you.